So it's running from 12 till 2 today in this order, and you can see there we've got a real range of topics, some of which feel, I guess, relatively well known in men's health, and some of them are far less well known. And then at the end, we've also got a lived experience by a patient advocate who's going to talk to you about their personal journey. So firstly, I'm very delighted to invite Professor Joris Feldman here today. So Joris, I think, has got lots of hats, and there's lots of things I think he could talk about in men's health, but he's got a very specific focus today of male infertility and genomics. And he's representing the University of Edinburgh, where he's the chair of reproductive genomics there, and also the director, Institute of Genetics and Cancer. Joris, would you like to share your screen?
Yes, I will.
That's perfect. That's worked.
Looking good in the right presentation mode. Yeah.
Perfect. Yeah.
All right. Thank you so much for this invitation to speak here, Becky, and thanks to all of you joining. It's a real pleasure to present this work on infertility genetics because it needs much more attention, and I hope to make it as accessible as possible for everybody on the meeting. So let me just start sharing now. Yes. So what I thought of doing is just briefly starting with a little bit of an introduction on genomics for those of you who are less into genetics, because it's really important to paint a picture of where we are in the field of medical genomics, genomic medicine, and then see how this is applied to male infertility genetics. So clearly a lot has happened in the field of genetics in the last decade or two. I've been very much involved in this, initially from the Netherlands and later in Newcastle and now in Edinburgh. And basically, we've moved from genetics being a approach that was very complex, and we used very many different techniques to study a very small part of the genome, to basically going to a single test where we can now look at the entire genome in one go, and it's even become affordable. And that's all because of miniaturization and parallelization of the system. It's a little bit like the computer system. And this has really made genetics much more at the forefront of medicine. And I gave my inaugural lecture just 2 days ago here in Edinburgh, and I talked about basically genetics being a starting point in medicine more and more, because it helps clinicians to guide not only, let's say, knowing what is happening, but also to guide therapy, and that's fantastic. And of course, the interesting thing is that we can basically go to now a single test genetics, where we take some DNA from blood or saliva of a patient, do a little bit of work, get it into a big machine, and then get the entire genome out. And that's, we're basically talking here about 6 billion letters that we can read. We can look at all types of genetic variation, from small point mutations to entire chromosome abnormalities. And we can do that quickly, and we can do that affordably. So that's very exciting, of course, for geneticists, but it's also really, really what is changing the field of medicine. And that's not just because I say so, but you can see that if you look at literature, and the UK is very much leading in this field. One of the reasons why I moved to the UK in 2017. So you see here just a few papers from the last few years where routine genome sequencing is being done in clinical settings, so not in research projects, but in big clinical settings. So that's very exciting for rare disease and cancer. So that was just a starting point. But now let's look at male infertility genetics. So we will hear more about Klinefelter syndrome later from Peter Marks. But yeah, that's one of the very well-known syndromes associated with male infertility because of this extra copy of chromosome X. So that's very well known, was identified a long time ago. And then also more than 20 years ago, small deletions on Y chromosome were detected as a frequent cause of severe forms of male infertility. And basically, this is almost where we've left it in this field. So we know a lot about chromosome abnormalities on the X chromosome, we know about the Y chromosome, but basically we know nothing about the most common forms of genetic variation, and those are mutations in individual genes. Those are completely missed and understudied in this field in diagnostics. So that basically means that the large majority of our patients do not get the proper care in genetics that other patients with rare disease and cancer will get. So it's just not fair, I would say. So this is just, again, this overview. The Klinefelter syndrome was identified in 1959. The CFTR gene, which is a gene specifically associated with obstructive forms of male infertility, was discovered in 1989. And then these deletions on the Y chromosome were detected in 1995. So these three have been implemented into routine diagnostics, not even everywhere, but that's at least mostly what is a kind of a minimum screen. But since then, very little has happened in the field. And that's strange, and especially for me as a geneticist, when I came from a different field where basically genomes are now routinely being implemented. So a main question that I want to address and ask you and get help with is, why is this field of reproductive genomics so behind? It feels almost that there is no need to understand the cause of infertility. And it is absolutely true, and I show some examples, but the field is understudied and underfunded. Patients' involvement is very minimal in our research. Clinicians, to be honest, also are very little involved. They basically, most andrologists would look at an infertile couple and say, let's try IVF or ICSI, without thinking what actually is the underlying cause of the infertility. So is it perhaps not important? Is it because we are dominated by private clinics and less by academia? Is it perhaps because IVF and ICSI have been so successful, or at least are thought to be so successful? And of course, we have to be realistic here. IVF success is not that high. It's about 60% after 3 cycles for women under 35. So it means already in the most fertile group of women, 40% will not become pregnant, let alone for older women. And basically, we often ignore the man a little bit and ignore what's happening with him. So that's not good. So I've been involved with a number of initiatives to try to both raise awareness, but also bring the field more together. So an important initiative is the Male Reproductive Health Initiative, which is basically a collaboration of colleagues around the world to try to advance the field, very much to basically raise awareness and obtain more funding for the research. Similar, more research collaboration, AndroNet, and then a collaboration, an international consortium which I helped to found, which is the International Male Infertility Genomics Consortium. And that is a consortium where we basically sit together with geneticists studying male infertility, and we share as much as possible data around that we find in our patients. Because similar to other fields, this is not a field where you can basically solve everything yourself. You find mutations in very many different genes, and you basically need to share these with your colleagues to find second or third patient with a mutation in the same gene, to learn about that, to learn about the clinic observed in this patient, and that's really critical. But also to link geneticists to biologists who can then do functional studies to learn more about what do these mutations actually cause. So this is something we've been very much involved in. And just a few slides to show some of the results from this. So this is from the Male Reproductive Health Initiative, and basically a survey was done amongst infertile men, and looking at various questions. And what is very obvious from the answers is that a lot of patients feel quite negative about their condition. Do not likely talk about it with others, do not know whether there's support groups in their country. And another one here on the bottom right, if you look at it, is that it asks whether an infertility specialist has recommended herbal supplements as your treatment for infertility. And you see that is happening in about half of the cases. And of course, herbal supplements may have their value, but of course, if you're an azoospermic man with a genetic cause of infertility, a herbal supplement will do nothing. And I think it's just quite shocking that we know so little about these men, and we just start giving them all kind of things. So it's a field which is, I would say, in the dark ages when it comes to understanding of this. When you look at the funding for this research compared to other fields of research, similar picture is painted. So you see here that if you look, for example, in the UK, and this is just for, I think, for the Medical Research Council, then about £1 billion is invested in research annually. And if you then ask how much of that research goes to male reproductive health research, it's about £2 million, right? So that's 0.17%. So this, of course, means that we can just basically do very little to advance this field, and that's again something that really needs to change. And this is the country that invented IVF, right? So I think we need to do more. So what's the consequence of this lack of understanding? Well, first of all, if you don't know what the causes of a disease, then you basically treat the disease as a common group. So basically, if you cannot, let's say, identify subgroups, so if you just say this man is infertile, and then you know, for example, that he has azoospermia, you just treat all men of azoospermia the same. Whereas if you would know that a man suffers from a specific subtype, a genetic subtype of infertility, and you know that, for example, that genetic subtype is associated with a successful IVF or ICSI outcome, then actually you can treat men and couples based on individual aspects. So this is what we call personalized medicine. And at this moment, that's not happening for male infertility. So we don't know if an assisted reproductive approach would be successful in an individual couple. We also don't know whether even if we are successful with assisted reproductive approaches, whether we would pass on the infertility to the next generation. So genetics is very useful for that, because it not only tells us what the causes of the disease, but it can also tell us what the inheritance pattern is. Basically, is it a recessive inheritance? Is it X-linked? Is it dominant? Or is it, for example, polygenic? So there's all kind of different types of genetic abnormalities. And if you have a dominant cause of infertility, it basically means you have a 50% chance of passing this on to your offspring. So that would be useful to know. That's something that, again, in other diseases, we know and we test for. So, for example, if you have a child born with intellectual disability, and as a couple you would like to get pregnant again, but you're worried that you get another child with intellectual disability, actually knowing that your first child has a dominant form of infertility because of a new mutation tells you that there is a very low risk that the second child will get that again. So those informations are really important. We also know that infertile men suffer from more health issues, and it would be really good to understand that better. And again, genetics can help with that, because you can then individually study genetic subgroups, look at associated diseases such as prostate cancer or other diseases, and that can help us to provide more information and prevent some of these diseases, perhaps. And we also know that for certain genetic subtypes, actually, age, so basically becoming older, has an effect on sperm production. So again, you could give advice to these men. And of course, the less we know about the causes, we can also not develop treatment options to improve things. So again, through these initiatives, we're trying to raise awareness. So this is another publication from this Male Reproductive Health Initiative. And yes, at least as a geneticist, I'm pushing for genetic causes to be added to the topics that need to be studied. Of course, there's other areas that we need to study as well in male reproductive health. And also, what is very much, and as it says here on the top point number 5, is that kind of integration of exome and genome sequencing, so these really modern tests, into the diagnostics of male infertility. So it's really important that this doesn't stay something which is just done at the university level in small research projects, but actually that genomic test becomes integrated and part of the NHS routine testing, because that's the only way where we can do these things at scale, and we can learn from that. And we can diagnose a considerable amount of patients, but actually also, again, benefit from this research. So we're pushing for that. I've written another comment in a journal last year together with my colleague Frank Titelman, also pushing not only the clinicians in the andrologists, but also geneticists like myself and clinical geneticists to care about male infertility more, and basically explaining that infertile men should be studied also more in genetics. So all of these things we're trying to promote. But then going back to the research, if you would like to make a link between a disease and a genetic component, one of the critical aspects is actually standardizing the way the clinic, the patient description is done. So basically the phenotypic classification. So when we study a genome and we find a mutation, we have a very standard way of describing that mutation. However, clinicians around the globe describe infertility phenotypes very, very different. And that makes it very difficult then to associate genetic abnormalities or findings with a specific clinical phenotype. So this is where the human phenotype ontology has been developed also and applied widely in other diseases, but was not yet used in infertility genetics. So this is what we published last year, an example of where to more systematically and standardized description of the phenotype, which then helps if you find genetic abnormalities to link these to these specific phenotypes. So that's critically important as one of the things we did. These again are just some examples to explain where genetics can help fertility clinics and help to improve patient management and providing information to couples. So just as an example, if you have a deletion on the Y chromosome, so here in the middle, the AZF deletion, then we know if you have a small deletion of just the region around AZFC, that actually you can still be successful in finding and identifying a sperm that can be used for ICSI procedures. However, if you have a very large deletion of the entire region of the AZF region, there's basically no chance that you can find sperm. And so you know this at the start. So you can basically, by doing a genetic test, you can already say, okay, this is what we can prevent. We should, the couple should be informed that basically it's not possible to get children through this procedure. With CFTR mutations, this is associated not only with infertility, but also with cystic fibrosis. So it's important to look at both the male and the female partner to look at a risk for CF in offspring. Then there are mutations, again, for which we already know that if you have a mutation in for example, the AURKC gene, you would basically block meiosis totally, and the sperm, even if you find sperm, it will not be suitable for reproduction. Then there are mutations associated with familial breast cancer and ovarian cancer, so that's important to know. And then finally, one of the new genes which we identified together with colleagues, once we started looking at the clinical outcome in patients with mutations in this CFAP47 gene, we found that ICSI procedures actually are very successful in affected couples. So that's actually really good news. And because this gene is located on the X chromosome, and if we pass on this mutation, it actually would be passed on to a daughter and not to a son. So basically, the mutations will not be passed on to male offspring. Of course, the daughter will then have a chance to get this mutation. So again, you need to keep an eye on that. But it would not result, if there's a boy being born, it would not contain these mutations. So in other words, there is good reason to continue on this because there's so many genes and other genetic abnormalities still to be identified. But we did one analysis a few years ago where we basically went through literature to look at all the literature in which genes were associated with male infertility. And you don't need to go through this, but basically to let you know, we've done this very systematically. We basically looked at what the evidence was that the genes reported were really associated with male infertility. It should be included in genetic tests. And what we found is that for a large majority of genes, actually the literature was very poor, and there's only a small number of genes that have let's say, enough sufficient evidence to be associated with male infertility. So you see them here. And again, if you would click through that, there's actually just a small number of genes which is associated with the more common forms of azoospermia and severe oligospermia, which we see in the population. So still a lot to be done. These are a few genes that we've identified through our International Male Infertility Genomics Consortium. So again, as I mentioned, this is where our group or another group finds mutations in a gene in an infertile patient, and then asks basically the rest of the consortium, do you also have mutations in the same gene? And in this way, we've described a number of genes because we could better characterize these and study these. We also sometimes could actually see that some genes reported by other groups were not associated with male infertility, so very useful. There is a number of groups in other countries who are now starting to implement these kind of larger scale genome tests called exome sequencing into the clinic. I think these are publications from Estonia, from Germany, and from France. And you see that they are starting to use these approaches, and they do have a reasonable diagnostic yield. So they found between 8 and 20% of diagnoses. In addition, they found many new interesting genes that could be followed up in research. These are still very small studies, but it does show that other countries are going there. And I also know from my colleagues in the Netherlands that they are basically doing this now in a clinical setting. So this is slowly but surely happening. So finally, just a few minutes on research we're doing in Edinburgh, and I've done in Newcastle before, because this is something about a specific type of mutations that we're interested in, which have so far not been studied in infertility, and these are called de novo mutations or new mutations, where basically you get half your genome from your father and half your genome from your mother. But if you would compare your genome to that of your parents, you will actually find that you will have about 100 new mutations in your genome. And these are associated, that's in general how evolution works. So we all have these, and this is how variants enter our germline. And if they are neutral, which most of them will be, they will be passed on to next generations. But these mutations also play an important role in genetic diseases. And our interest was to study whether they could also play a role in fertility. So as I mentioned, we've previously studied these in rare syndromes and intellectual disability, and now we're studying these in infertility. So basically, a group from America mentioned that a point mutation was identified once in a patient with infertility in 1999, but that's the only study so far. However, we know that Klinefelter syndrome, as well as these deletions on the Y chromosome, are both new mutations. So they may play a more important role than we think. The problem, however, is that in order to study these new mutations, you need not only to obtain a DNA sample from the patient, but also from the parents. And that's perhaps not so difficult when parents come to the clinic with their child who has intellectual disability or developmental delay. It's much more difficult if an infertile man comes to the clinic, because he doesn't bring his parents to the clinic with him. So we are the only group in the world who does these studies, and we've been working with patients who've been kind enough to allow us to contact their parents and obtain saliva samples from parents in order to compare the genomes of infertile men to that of their parents. And we published a paper in 2022, a first paper where we showed that these new mutations are very important in infertility. We've used different approaches. We now have more than 300 patients and parents that we have done genetic studies in. We're writing up different papers, but what you basically do is you compare the DNA of the patient to that of the parents, you filter out the inherited variation, and you can then look mostly at the new mutations using all kinds of computational approaches. You can then see all kinds of interesting things, which I will not go into detail at all today because I'm already running out of time, I think. But for example, we see a lot of mutations in genes involved in RNA splicing, which is very interesting. Through collaboration with our colleagues in the International Male Infertility Genomics Consortium, we then find a number of our genes also mutated in other cohorts, and that again then puts these genes as top candidates to further study. So that's something we're doing. And I'm now starting up my research group here in Edinburgh with colleagues, Margot and Alex, who have joined me, and I'm recruiting a PhD student and a postdoctoral fellow to further join me, basically to improve the understanding of genetic causes and make sure that we get this implemented to the clinic. Edinburgh has a Centre for Reproductive Health, so there's a lot of understanding around the biology of reproduction, a lot of oocyte biology, so female infertility as well. However, we need to do something, so there's a lot of work to be done. So more genetic sequencing, more genome sequencing in infertile men and their parents. We used to novel approaches, going from short to long read genome sequencing, where we can detect more variation, following up the genes we identify, both functionally and clinical, and basically expanding from male to female infertility genetics as well. Finally, I think, infertile couples deserve better than the kind of genetics testing that they get now. So how can we do this better? I need help. So please, there's 88 people on the call. If anybody wants to help, let me know. But one thing that I've started is a charity because we have patient organizations that very much support infertile couples, and some of them may be on the call, to basically improve support for these couples, which is critically important. But in contrast to other diseases, there are no charities that really support research in this field. And I'm a director of an Institute of Genetics and Cancer, and my colleagues in cancer research, they have hundreds of charities, including Cancer Research UK, which is the biggest charity in the world. So therefore, I decided to shamelessly copy that and make it a charity called Fertility Research UK. This is all very new. You cannot donate yet, so it's only just starting. But if anybody's interested in supporting me and helping me with setting this up, please contact me by email or else afterwards. So thanks very much for everybody on the call. Of course, this is work that I've done with many colleagues throughout the years, and it's lots of collaborations. It's a very exciting field. This is a field where, on the one hand, we're only just starting, and on the other hand, we're already making lots of clinical impact. And happy to answer any questions later. Thank you so much.
Thank you so much, Joris, for that. I think you've highlighted some really important issues there. It's not so often we hear about male conditions that don't get the funding. I think I should say that as the only woman speaking today.
Fertility Research UK is also for female infertility, so it's for
Okay,
everything
that's great.
related to research. Yes.
Thank you. And yeah, I can only imagine the challenge of trio testing for adult males as well. I think a lot of people on the call will kind of emphasize with that. So thank you so much. And yeah, please do put questions in the chat, and we will definitely try and get to them at the end. And if you want to share any of those links in the chat as well, Joris, please feel free to. That would be great. So I'm just going to skip forward a little bit to our next speaker. So our next speaker today is also obviously covering an important male health theme. So it's Peter Marks. He's here in Birmingham with me at Birmingham Women's and Children's Hospital, and he's a genetic counsellor. I should add there's probably lots of other things I could have also asked him to speak about, but this is very topical for our talk here today. Peter, would you prefer to manage your own slides, or it's up to you?
Yes, I'll give it a go.
That's great.
How it goes. Is that okay?
Yep, it's loading. Yeah.
Is it in the right view?
Yeah, that's right. Perfect.
Great, thanks. And thank you very much for inviting me, Vicky. I'm delighted to be able to speak today. And it's quite by serendipity that my talk follows on from Joris's with the mention of Klinefelter syndrome, which will indeed be the focus of my talk. But I'm also hoping that along the way, maybe we can think about challenging our perceptions of the X chromosome, of gender identity, and the role of testosterone in men's health along the way. So I hope it will be interesting for everybody. Just to introduce me, I'm a consultant genetic counsellor here at Birmingham Women's and Children's NHS Trust. Now, I'm really fortunate to be able to specialise almost entirely in endocrine genetics, which really gives me a chance to be involved in with all sorts of interesting patient groups, from endocrine tumour predisposition to more general endocrine genetic conditions. And one of the groups and clinics I work in particular is differences in sexual development, and I'm privileged to work with patients, men with differences in sex chromosomes, and you may see this in the technical literature as sex chromosome anomalies, or SCA. Now, there is this sort of received wisdom when we think about sex chromosomes that the X chromosome is somehow a female chromosome and the Y chromosome is a male chromosome. But I suppose with the notion that in fact Y is the sort of sex determining chromosome, it occurred to me in the shower this morning, of all places, why do we even regard X as a sex chromosome at all? But that's sort of a more philosophical point. And the reason I talk about this is because the Y chromosome, for some years in genetics, we regarded it as not very important, obviously, for sex determination, but some genes on there, not many compared to the rest of the chromosomes. And once upon a time there was talk of a hairy ears gene on there. Obviously we know now, and of course Joris made reference to this, about the importance of the Y chromosome for sperm production. But I'm going to talk a little bit about Y and the sex determination pathway. This, I'm afraid, is a very simplified version of the human sex determination pathway, and you can see there that the SRY gene from the Y chromosome is sort of the primary sex determining factor at the top of the pathway, which sets in motion lots of other complex genetic expression pathways, but essentially it's what triggers the undifferentiated gonads to develop into testes usually, and then that in turn triggers a sequence of hormonal changes that leads to the development of the internal and external reproductive structures. And then, so you can sort of see on the right hand side how we might regard people in terms of chromosomal sex, gonadal sex, phenotypic sex, and then finally there's a whole spectrum of factors that sort of influence how people feel in terms of their gender identity, and I don't think we understand all of these by any means. These are a list of some of the sex chromosome anomalies that we do see in men. And you can see Klinefelter syndrome there at the top. There's some variations in Klinefelter syndrome where we might see some additional X chromosomes as well. And then we've got all sorts of combinations where you might see a mixed population of cells, what we call a mosaic pattern, where you might have some cells having a single X chromosome, some cells having a Y chromosome, and I get to meet these patients in my clinics too. And then there is an XYY condition, which does have an impact on male fertility. Actually, it is can be somewhat reduced, but not to the degree that we see in Klinefelter syndrome. So this is where I focus in a bit more on Klinefelter syndrome. Now, if you look at some of the morbidities that we see in patients with Klinefelters, it can obviously look like a huge multi-system disorder, and they're the symptoms that you see with a red asterisk next to them are those that potentially can be modulated by testosterone replacement therapy, which I shall come back to. But you can see that a lot of those are really secondary to short or long-term testosterone deficiency in part. So a few facts about Klinefelter syndrome. It affects up to 1 in 500 boys and men, and we think it's still very much undiagnosed in some men. And there are chiefly three points in life when Klinefelter syndrome can be diagnosed. The first point is antenatally, so perhaps somebody's having a chromosome test in a pregnancy because of maybe some anomalies that have been spotted on scan, for instance. And at this point, we would regard Klinefelter syndrome as what we would call an incidental finding. So it's very unlikely to be the cause of any abnormalities that you might see on an antenatal scan. And so really this hits couples very hard, because not only do they have some abnormalities discovered on scan, which potentially could be very serious, they've also got this incidental diagnosis. And it might even be the case that perhaps some of those anomalies on scan resolve along the course of the pregnancy. And so you might find that couples are left with just the Klinefelter syndrome as a diagnosis to think about, and leaves people a little bit bewildered as to what choice they might make around that. And we certainly know that some people have felt strong enough to terminate a pregnancy on the basis of that result, although we don't see it very often. The next point at which somebody might be diagnosed is in childhood. And again, it's usually not diagnosed because somebody looks like they have features of Klinefelter syndrome, although we do know that that happens more often than not. A child may be investigated because of some developmental or learning disability, and they're having sort of generalized chromosome testing, which is usually a detailed test called a chromosome microarray these days. And so it might actually be that a child ends up with this diagnosis, and it may indeed not be the underlying reason for the learning disability that they've actually presented with. And we do need to watch out for attributing a child's learning disability to Klinefelter syndrome, because it may be more profound than we would expect to see in Klinefelter syndrome. And I've certainly known patients where they've had Klinefelter syndrome, for instance, identified on a karyotype, and then I felt that their presentation in terms of learning disability is actually more than we would expect to see from Klinefelter syndrome, and then actually further genetic tests have revealed an additional cause for their learning disability. But obviously this is a huge impact for parents, and I'll talk about a patient that I came into contact with last week to try and give you a bit more insight into that. And then finally, and this is really where a large number of men are diagnosed with Klinefelter syndrome, it's where they're presenting in fertility clinics. They've had multiple investigations looking at some of the genetic causes that Joris mentioned in his talk. And then they're identified because of azoospermia as having Klinefelter syndrome. And obviously this, again, is devastating for somebody to receive this diagnosis. And in many ways, it's devastating at this stage because there is the lost opportunity for testosterone replacement that could have been so much, given them so much benefit for their health and possibly mental health at that point, as well as coming to terms with the fact that it's obviously the cause for their infertility, and the age at which they present might mean their options are more limited now for treatment. So the diagnosis can be devastating at whatever life stage. But in fact, we would generally argue that childhood diagnosis is probably the optimal time in terms of treatment, because you can get that testosterone replacement started, if required, at the appropriate point in life. But the vast majority of people with Klinefelter's are diagnosed towards adulthood or in adulthood. Now, what is Klinefelter syndrome? Now, obviously, we've talked a lot about it, and we've talked about it being an additional chromosome, but part of our job as genetic counsellors is really to sit down with patients and families and be able to communicate what is often complex genetic information in a simplified way. Now, since I've been working in endocrine genetics, I've really thought very carefully about how I communicate about Klinefelter syndrome and what is the simplest possible way that I can get across this information. Now, hypogonadism is a very medicalized term, so actually I don't say to my patients, actually Klinefelter syndrome is essentially a form of hypogonadism. What I will tell them is that the testes don't develop and grow as they should do at puberty. And that essentially is the headline that I'm trying to get across. And then I can say, well, testes have two main functions. One is to produce testosterone, and the other is sperm production. Then we can get into a conversation to address both of those aspects. So we talk about the short and long-term benefits of testosterone replacement therapy. And then on the flip side, on the other side, we can talk about the implications for fertility and what they may be. And then, of course, not forgetting, especially if this diagnosed in childhood, thinking about the specific neurodevelopmental features. And what I mean by that is we know that in particular, men diagnosed with Klinefelter's are often, well-functioning men with jobs. They've probably struggled to some degree at school. They may have had some speech or expressive language issues with development. Very often, in my experience, they've encountered difficulties with socializing, making friends, have problems with self-esteem. So it's those kinds of things. So again, this is a very simplified pathway. Now, in endocrinology, we love to show loops and cycles and feedback loops and how they work in the body. This is a very simplified diagram. I like simple, as you might be able to tell by now, as you can tell from the source as well. But essentially, it's the hypothalamus that sends the signals to the pituitary that in turn stimulate the gonads, and we're thinking here about the testes, to be producing testosterone, which should be acting as the brake or the negative feedback factor, acting on both the hypothalamus and pituitary, which says, okay, we're functioning fine, let's slow down those stimulating factors. And that's the whole basis of the feedback loop when it's working correctly. Just to show this in a little bit more detail, here's the two functions of the testes. You have the Sertoli cells stimulated by the signal called FSH, the hormone called FSH, which are working to produce the sperm cells and maintain them. And you've got the Leydig cells there producing testosterone. So again, it's nice to come back to some of these medical terms and think about what they actually mean. And when we think of hypogonadism in Klinefelter syndrome, we talk about it being a primary hypogonadism. This means the testes simply are not functioning, no matter how much you might stimulate them from the pituitary gland. And so this leads to the very characteristic pattern hormone profile where you have elevated gonadotropins, which are these FSH and LH hormones that are released from the pituitary. And of course, you see that usually a testosterone deficiency. And then again, this helps us understand these rather clunky terms like hypergonadotropic, which means high levels of FSH and LH, and hypogonadism, where they're just not working or functioning. So, with the technical stuff out of the way, it's nice to be able to talk about an encounter with a patient's family last week that I had. So the patient referred to us is in fact a 9-year-old boy who was investigated under the Child and Adult Mental Health, the Child and Adolescent Mental Health Services for learning and behavioural difficulties. And just to give you a bit of context as to what was happening here, this is a boy with some difficulties at school, not significant learning problems by the sounds of it, but he was also developing some challenging behaviours, which sadly were mostly directed towards his dad at home. And we reflected together how it was probably because home and dad are very much his safe place and safe people. And those of you who are parents will know that as they're safe people, you often get the brunt of the more extreme behaviours. And so in this case, the child got a microarray, which was a detailed chromosome test, and it revealed the likely diagnosis of Klinefelter syndrome. The parents were informed of this diagnosis by a telephone call, and I have to say they haven't had any face-to-face support ever since that telephone call, which was 12 months ago now. Dad was the person I was talking to on the telephone, and we talked for about 45 minutes or so. And he just described it as being a devastating piece of information to find out about his son. He remembers very clearly that basically he was told it means he'll be infertile, prone to cancer, and blood clots. So these were clearly the things that stuck in his mind. And lots of things to come to terms with there, and especially in a 9-year-old, and then probably with this adult overlay of what it will mean to be infertile in the future, and thinking he also talked about the impact on the development of his genitalia, and thinking about how difficult this will be for his son in the future. So I reflected on the fact that there was no face-to-face support really offered for this family. So my instinct was to say, look, would it be helpful if in fact you came in to see me, you and your wife, so that we can have a discussion about it. I could tell they had so many questions that they wanted to ask about this, and I was really mindful that nobody really had offered that kind of support in the past. In the last couple of months or a few weeks ago, I think the family had actually ended up going onto a Facebook group, which probably was quite helpful for them. And I directed them towards some more online support as well. But the fact is that they ended up with a genetic counselling appointment 12 months after the initial investigations that this child had, and that really doesn't feel like ideal really for family to be getting some detailed answers to their questions so far along really. And I was mindful that it was a telephone consultation, and so I was very keen to offer a face-to-face conversation really, which is what will happen in a few weeks' time, I'm pleased to say. And of course their son's not aware of the diagnosis yet and the implications. And I think it will really help, as I often try to do with parents, is try and plan that moment or moments when this information will be shared in a way that's suitable for their age and understanding, really, and also making sure that he will get the opportunity to come himself when he's older and asking his own questions to genetics. But against that backdrop of, what is a devastating diagnosis in many ways, is what I tried to explain is that childhood is really the ideal time for the diagnosis. Obviously, antenatally, it's not a great time to be getting that sort of information. And in adulthood, you've missed out on that opportunity for therapy. And so childhood, pre-puberty really is the ideal time to be getting the diagnosis. We can make sure then that the child has pediatric endocrinology input at the onset or slightly before the onset of expected puberty, because puberty for people with Klinefelter syndrome usually will happen spontaneously. Some children do need help with that, but it's to really identify the moment at which a child becomes testosterone deficient, if they do, and then commencing the testosterone replacement, which will have all sorts of benefits. Obviously, the long-term benefits are there, but for heart health and the other morbidities, bone health. But then we think about the improved mood and musculature, which are going to be so important as well for a child's self-esteem, particularly as they go through puberty. And I've been working with some young men with Klinefelters who are sort of in their late teens or early 20s who really, I don't want to over exaggerate the effects of testosterone, but they've seen their life very much transformed by being on testosterone replacement therapy. It's really kind of turned their life around to some degree. And of course, in doing so, we hopefully reduce the likelihood of the longer term consequences of testosterone deficiency. And then as well, you can begin to think about what the reproductive options are. It's a little bit controversial as to when you might decide to think about what to do about potentially extracting gonadal tissue for sperm freezing or use in fertility treatment. Generally speaking, we don't have huge amounts of data around the success of IVF in men with Klinefelters, but we're sort of understanding that treatment before the age of 30 is going to have the most chance of success. The age at which you might try to extract tissue or try and extract sperm surgically, we're a little bit less certain of, and it's certainly going to be a tricky conversation to have with a teenager who won't be contemplating starting a family necessarily anytime soon, and so finding a way to have a conversation about that could be quite difficult. And I have to say, the success of therapy and, even long-term psychological outcomes, because I very often refer patients to a psychotherapist that we work with as part of our multidisciplinary team. All of this relies on reliable and consistent management, which I'm afraid to say doesn't exist universally. And although we do have a specialist clinic reasonably locally that looks after adult men with Klinefelters and tries to bring together a lot of holistic aspects of care. Very often the patients are cared for by local endocrinologists, and the keeping these patients really engaged with monitoring their hormone levels and their treatment is very hit and miss, we find. And I'm hoping that in the not too distant future, we will actually set up a dedicated Klinefelter MDT clinic here in the Midlands, along the lines of the clinics that have been established in London. so that's more or less what I'd like to say. I mean, I will come in with another thought about this, which is I recently heard a podcast, a rare disease podcast, where there was a patient with Klinefelter's there who actually identified as female and identified as female long before she was given the diagnosis of Klinefelter syndrome. And the most fascinating thing she said was that everyone who explained it to her said that it was an extra X chromosome that had caused her condition, but she would argue that it was actually an extra Y chromosome in her case. And my mind was actually blown when I heard this, because I actually thought, why do we say an extra X chromosome for Klinefelter when you could say it was either way, but that just adds a further dimension to the way we think about sex chromosomes, gender, and all those sorts of things. So yeah, I hope you find that as interesting as I did. And yeah, that's all from me.
Thank you so much, Peter. I wonder if you could share that podcast, actually. I think there might be people who might be interested in that. I think that sounds really interesting. And I'm surprised actually that a 1 in 500 doesn't actually feel that rare compared to like possibly how well known this is in the general population. So yeah, thanks so much for sharing. And yeah, of course, please do ask any questions and we'll try and get to them at the end. And I'm hopefully, Julian, are you available? Are you here
I
somewhere?
believe I am. Yeah.
Brilliant, wonderful. That's very encouraging. Let me just find your slides. Would you like me to share them, or would you prefer to?
Well, I will see if I can do it from here.
Great. Okay.
But if not, it's always good to have a plan
Amazing.
B.
Yeah, certainly.
How's that? Is that coming up?
Yeah, brilliant. So I'm delighted to also invite Julian to talk now. Thanks so much for coming. So Julian's based in the University Hospitals of Leicester, and he's a clinical geneticist. And again, I think there's lots of things that he could speak about, but I'm delighted that he's going to be talking about prostate cancer today. I think it's something that we feel that we talk about a lot, perhaps as a society, but perhaps not so much from the sort of genomic angle. So thanks so much, Julian. I think you might need to just put your screen full, perhaps.
Yeah. Yep. We will do that. And brilliant. Thank you so much. Is
Yeah,
that
that's
coming
great.
up?
Perfect. Thank you.
Fantastic. Thank you. So let's check this all pushes through. Fabulous. So just a word from some of my sponsors. So thank you so much. So when I think about the difference of genetics and genomics, so one is the why, what, and the when. And genetics is really classic about family. So how did we get to this particular point? So our oldest gene is 2 billion years old. So it took 2 billion years for us to get our bodies. And to be honest, I think I want my money back if this is all we could come up with after 2 billion years. But anyway, it's been a real journey, and genetics explains how we got to this point today. Genomics really is bringing different types of data together linked with healthcare outcomes to make better predictions. So genomics for me is about when, and it's not just about subtle changes in your genes and in between the genes, and it's not purely just about looking at tumors or looking at cell-free DNA or urine ostics. It's about bringing all of this material together. Remember that your DNA code is only as powerful as your postcode when it really comes to predictions in the vast majority of individuals. Obviously, those with single high risk genes like BRCA, it may be a little bit different. And the intersection here is about individual patients. So this is things like looking at individual tumors, and we talk about treating disease for what it is rather than purely just what it looks like. We've obviously all been through COVID, and we know a lot more now about viral genomics as well, and looking at the characteristics of the virus as well as the host in predicting response. And pharmacogenetics, making sure people get the right drug at the right dose first time, every time. So I think it's important to be clear that there are some differences between those three when it comes to thinking about how best to support patients and populations as well. So when I started in clinical genetics back in 2001, this is pretty much where we were at when it comes to looking for genes. So we've got the classic BRCA1 gene. So Angelina Jolie had preventive breast surgery and reconstruction. And I always hoped she changed my life. It wasn't quite what I had in mind when it tripled referrals locally, doubled nationally in about 2014. And we wrote a paper you may have heard of called the Angelina Jolie effect. Now, when it comes to prostate cancer, classic gene BRCA1, but actually the main risk associated is BRCA2. And here we get male breast cancer as well. Men have breast too, BRCA2. And remember that these genes can pass through the maternal, but also the paternal side of the family. And prostate cancer tends to be at an earlier age. It tends to be more aggressive, and it tends to be more common as well. So when we see these things, we always look for genes running in the family associated with breast and ovarian cancer. You can see TNT, so triple negative breast cancer as well, breast cancer and ovarian cancer. But in BRCA2, we also see a lot of bilateral breast cancer, male breast cancer, gallbladder tumor, the cholangiocarcinoma as well. And there are a series of these links. So breast, ovarian, pancreas, and prostate for the BRCA and PALB2 gene. The bowel, gynecological, upper GI, and prostate cancer, particularly MSH2 in Lynch syndrome. But we see kidney tract tumors and epididymal cyst tumors in von Hippel-Lindau as also an association with brain tumors and neuroendocrine problems. And then you've got melanoma and pancreatic cancer can be linked to the cell cycling genes, breast and stomach with E-cadherin, breast, thyroid, and uterine for PTEN as well. And we've written a book called Clinical Genetics and Genomics at a Glance for those that would like to know more about some of this inherited types. So a classic type is kidney cancer, which can be linked to a number of different syndromes, but also chromosomal problems as well. And in clinical genetics and genetics at a glance, one page is writing and the other page on the opposite side has complementary pictures. So hopefully you read it and then can find out more information about it through the pictures as well. So the story for genomics in terms of familial cancer really starts here, when classically we started looking for BRCA1 and BRCA2 in patients with ovarian cancer. But then we found that if you extend into wider gene panels, rather than finding perhaps 10 to 12% of patients with gene changes causing inherited ovarian cancer, actually there's other genes involved as well. So now, rather than looking at individual genes, we are often able to look at a wider panel, and that increases our ability to make a diagnosis. And when the 100,000 Genome Project started, we had 3 sisters with breast cancer who were diagnosed with breast cancer within 13 months of each other. And we were able to go onto the BBC Breakfast Sofa to explain the project in terms of looking at the whole genome, all 6 billion letters, and comparing patients to see if we could make a diagnosis. And this was also not only looking at inherited risk, but looking at tumors as well. So we went on the BBC Breakfast Sofa, and then again, when we found a new gene in the PALB2 in some of the family as well, and we're able to do further predictive testing as well. So that was a very important message about being able to develop new databasing and also test tumors as well. So we can treat disease for what it is rather than purely what it looks like down a microscope. What I call the Death Star analogy. You can't destroy the Death Star until you've got the plans. So this is getting away from purely looking down a 500-year-old microscope to actually trying to understand the weaknesses within the tumor. And of course, when you're looking at the whole genome, you might find other things as well that might be relevant to the disease, but may tell you other information. It's a little bit like a whole body MRI. So anybody having a full body scan tend to find 1.5 to 2 on average subtle variants, which may be important, but may not be. So consent is very difficult. We've got 20,000 genes, and we know of at least 7,000 rare diseases, 2/3 of which are caused by genes. So we know it's difficult to consent when there's so many different things that you can find. And there's a lot of issues about the balance between complexity and uncertainty, as well as how our data is stored and used. So consent is very important when you're looking at these broader testing and making sure that patients are on board. And that's going to be important when we consider, for instance, the generation study, which is taking over potentially from the heel pricks for newborn, looking at neurometabolic problems in childhood, but also potentially patients reconsenting to look at their whole genome data when they become adults as well, as we move to more population screening for inherited disease, and also looking at the subtle variants, which may be important for clarifying future risk of other disease. Now, if you had 4 minutes and 4 seconds left on the planet and didn't know what to do, I would urge you to go on to a very nice video on YouTube called Our Genome in a Circle. Now, you may have seen pictures of chromosomes and karyotypes, but when it comes to tumor genetics, we now represent tumors as these circles with the chromosomes, which are the packaging for our DNA. And you've got 22 pairs, standard pairs, same in men and women, then the X and the Y, which we've been hearing about today. All around the outside, the subtle changes are going closer towards the middle, here in red and green, and then the rearrangements between the chromosomes. So when chromosome 1 binds with number 2 or number 3 or number 4, what I call the Morecambe and Wise sketch, the Andre Previn sketch, got all of the right genes, but not necessarily in the right order, from that great sketch from the end of the 70s. So here we have an example of one of the families with PALB2, a breast cancer gene. You can see lots of chromosomal rearrangements, and this is what we call genomically very abnormal. But at a single base level, looking at the letters A, T, C, and G, there's actually not a lot of mutations here. So in the future, we'll be able to look at these wheels, which look at our whole 6 billion letters, and get a visual representation. And of course, with AI, you can then look at which treatments are most likely to be helpful. And these develop what we call genomic signatures, and we'll talk a little bit more about it. So here we have a Lynch syndrome womb tumor, and you can see, if you compare the two, the red and the green around the outside are much thicker, but in the middle bits, the rearrangements are much smaller. So you can see that from a genomic perspective, even at just first sight, these types of tumors look very different. And that's why it's really important that we are aware and we personalize the treatment based on the actual changes within an individual's tumor. Now, another thing that's developed, other than looking at the genes that we've inherited, is to actually look at the changes which in the genes within the actual tumor itself, the mutations, the changes in the genes, which may be important in driving the tumor. Now, we know that tumors are genetic, they're caused by changes in genes, but the vast majority of which are random, and they are just there and they've occurred as the tumor develops. They're not inherited. And also most of them are what we call passengers. They're not actually driving the speed of the growth of that tumor. And so the majority of those changes are actually occurring as the tumor develops. And I don't know if you've ever seen the Wizard of Oz, but the Wicked Witch gets Dorothy in the castle, and she has this spectacular egg timer with pink sand, and she flips it over and says, "You've only got this much time before I get those ruby slippers." And all the gene changes that we've had through those 2 billion years are in the top side of that egg timer. But over time, we develop new gene changes within our tumors, and that is the egg timer going back down into the bottom of the hourglass. So what we're looking at here is a series of genes along the Y axis. On the other Y axis is the number of patients that they're looking at. And then the red and the blue is the proportion of times where when we find a change within the tumor, is it likely to be actually inherited? So when it's all red, that means that those gene changes are very rarely inherited. Whereas if they are blue at the top of the page, then if you find them in the tumor, then they're likely to run in families. And the relevance and why I show it is by testing the tumor, you might be testing the family. And in life, when you're taking a decision, you need to ask yourself 4 things. Do I need to make it now? What's called the Falkland effect. Is it important and going to be required now? The second one, is it important? Is this information going to actually change anything? Thirdly, can you influence it? Finding a gene isn't any good if you can't change the treatment, or you can't work out who's at risk in the family, or if you can't offer screening or lower the risk. So you need to ask yourself that. But crucially, when it comes to these tests, it's also, is it reversible? And I always say any decision where you can't unknow it, you do need to give it a little bit more thought as well when you're going through. So it isn't just a matter of, well, we're doing this test potentially to help you in terms of treatment. It may be important for the wider family. And this is important also based on different ages as well. And Claire Turnbull is one of the very best in cancer genetics in the country at the Royal Marsden Institute of Cancer Research, who generated this data. We've gone on to support patients with PhoNaps, so that on a single view, with a red, amber, green system, you can see if you're screening for Lynch syndrome, aspirin, whether or not you've had a breath test, spoken to your family, gynae review, and symptom aware. So we are developing this as a forerunner for when you're into Boots. Other pharmacies are available, when you can track your prescription against how you're likely to respond to it genetically, because 1 in 7 prescriptions are incorrect because of side effects or the drug doesn't work. Now, I'm rather partial to Mr. Kipling, so being recently placed on a statin, and I have had a genetic test to see how I'm likely to respond to it. And we want patients to be able to feedback their experience of the drug. Did it work against their pharmacogenetic profile? So you can have a personalized Amazon style rating for your prescription on the high street. And this is taking genomics right into the high street. So if you've got cancer, your painkiller, your antibiotic, your blood thinner, your statin, whatever it is, will be your blood pressure pill, will be at the right dose. And these are very common genetic variants. So the familial cancer genes are only 1 in 100, but 99% of us have got a variant in these genes. And there's basically no such thing as average. So we're working towards it. And this is how this type of drug might potentially work. When it comes to prostate cancer, we test people based on family history, but we're also now, and we've developed testing where you can actually look at the prostate tumor as well, and to see whether or not we can find a gene change in BRCA2. And that's really important because there are new treatments now which target those BRCA genes, as well as telling you why the tumor has occurred. Are you at risk of male breast cancer as well? And also, is there a wider risk to your sister, to your mother, to your daughter of developing breast and ovarian cancer? So we've helped drive through testing of tumors as well as testing for family histories as well. And the reason why these are targeted drugs is the genes, the PARP inhibitors, they actually target the weakness of BRCA deficient tumors. So we've all got 2 copies of our genes, And what the BRCA genes do is they help cells when there's a double strand break in the DNA. And the way I like to explain it is it's a bit like if you were to explode Tower Bridge, how would you know how to repair it with the cantilever and the two towers? Well, you would need the plans. So the BRCA genes bring along the plans when DNA is damaged. So within the tumor, which has lost 2 copies of the gene, and the way I like to think of it, it's like having 2 copies of a book in the library. 1 copy has already been lost because it's been inherited, but within the tumor, you lose the second normal copy of the gene. And so this is a much kinder treatment for the body. So what you're doing is you're destroying and allowing the body to heal itself by inducing these double strand breaks, which within the tumor can't be repaired. It's important you try and treat these early. And maybe one day we'll get to a point, if you've got these genes, that for 2 weeks a year, you will go to a BRCA spa and you will have these drugs to mop up cells as they're developing a tumor. So the earlier you can treat people, then actually the more likely you are to be able to stop the individual developing an aggressive tumor over time. We've done a lot of work in the community. I want to acknowledge 2 ladies who are sadly no longer with us, who have done amazing work in the community, raising awareness. So Pamela Campbell Morris with the Play Dominoes Talk Prostate Cancer. If you want to talk to people in the community, you can't invite them to hospital. You've got to go out to the events where they are already happy and are prepared to talk. So domino events, particularly for people from Jamaica, and Rose Thompson, who did a lot of work with the Hear Me Now group and Check Things Out group, which went to Parliament and developed prostate cancer screening for men who in African ancestry, who are double the risk, 1 in 4 risk of developing prostate cancer, 1 in 12 risk of dying of their prostate cancer, and it tends to be earlier and it tends to be more aggressive. So being able to raise awareness for people in these groups who may struggle with thinking about prostate examination, and it is still a taboo topic in some communities, is incredibly important. And we are doing a lot of work campaigning to try and actually develop a national prostate cancer screening program for people of African ancestry, but also people who are very high risk because of these genes and because of their family history. So in the longer run, we need to bring different types of data sets together from our urine tests, and we've helped develop a urine test to pick up prostate cancer, particularly aggressive prostate cancer, because there is the rule of 15. So the problem is 85% of men at post-mortem, 85% of men at post-mortem over 85 have got prostate cancer cells. So it is normal to develop prostate cancer, but only 15% of men with prostate cancer cells die because of their prostate cancer. So it's more normal to actually die with prostate cancer rather than because of it. So what we need to do is to bring different types of data sets together, the urine tests, the PSA tests, the MRI tests, and work out who's got the early aggressive disease that we treat that will save lives, and without increasing the risk of lots of side effects like incontinence and impotence. So bringing data sets together is important, but there are challenges. You remember the issues with Horizon, with the post office. We know recent data breach, 23andMe. We know about the A-level algorithm fiasco in 2020, when the A-level results were given that were proportionate to your score, but also how other children got on in the previous 5 years at your school. So that's a matter of bringing your DNA code with your postcode. And the question is, at an individual level, it does impact on our sense of social justice. So I think people would be very worried about being screened based on their postcode alone or their ethnicity. So we do need to think about just the coulda, woulda, shoulda. And it's important we bring the social sciences and humanities with us when we think about introducing these types of programs. But at the moment, many men are dying in silence. Now, we know that prostate cancer screening from PSA is not perfect because, wait for it, 15% of men over 50 with a normal PSA could still have a significant prostate cancer. We know that only 15% of men will come forward for MRI and screening if you write to them cold as well. So all of these things need to be taken into consideration. And algorithms are fine, but it should not be a case of computer on and brain off. So as part of our United Against Prostate project, led by our fantastic manager, Joe Lowry, and working with Gemma Gunn and also Maria, our genetic counsellor with special interest in prostate cancer. And if you've got concerns, please reach out to us about prostate cancer in your family. Then we looked at these areas of African Caribbean ancestry and family history as well. And here's the rule of 15 as well, explaining why it is so difficult to get screening right. But of course, now with MRI, now with watchful screening programs, things have massively improved. And we feel now is the time is right to start thinking about supporting men at this very high level of risk as well. And this is all part of the process. We developed a new tumor testing pathway to be able to see if we can find those gene changes because of the rise of these new targeted drugs, as well as raising awareness in high-risk communities for screening as well. Because if you're going to show a benefit in screening, it's got to be in those who in balance are at the much higher level of risk. And this is part of the East Genomics GMSA. We did a video with Andy's story. Andy came out of the blue with prostate cancer, and we found a BRCA2 gene change in him, in his tumor, and then in his blood, and we're able to support his family. A very powerful video with a really incredible guy during the pandemic. So feel free to check that out. But we've done a lot of work with because we're united against prostate cancer, with our community groups, going out also to Leicester City Football Club, and the football club and the mayor were incredibly supportive in the Leicester region. But we went out throughout the region and very well supported by our patient public voice chair, Chris, and our patient champion, Eddie, as well, reaching out into the community, where other awareness events, also creative arts events as well, and radio broadcasts as well in targeted local radio stations as well. We work very closely with the Institute of Cancer Research, supporting research studies that are looking particularly at screening and treatments targeted for those with genes, but also screening for men from the African and African Caribbean community as well. And we're very happy to signpost people to the Institute of Cancer Research and the Royal Marsden for this. But we also go out and we do a huge amount of outreach and community engagement. My professorship is in engagement and outreach. We've done a book for children based on DNA, like what came first, chicken or the egg? Is granddad a dinosaur? Why don't I like Brussels sprouts? Why is there blue in a traffic light? How to spot a relative after 500 years? And if you don't know the answer to any of those questions, perhaps you should get the book. And I'm also transferring the book, would you believe it, into rap music as well, if you'd like a DNA rap at the end, if you haven't suffered enough. And a huge number of people have been involved in our work, both clinically and social media and classic media as well, into the creative arts as well. I'm very happy to share any information and contacts. We think it's important to have outreach, but also to have oversight from patient groups, insight from patient groups, as well as the really important aspect of lobbying, as well as stakeholder support for patients, and crucially also their wives and family, so that we treat disease for what it is, not what it looks like, that we screen people based on risk and not age alone, and we also make sure that people get the right drug at the right dose first time. Thank you so much for listening. Happy to take any questions from you today. Really appreciate all of your time. Thank you very much.
Thank you so much, Julian. I really enjoyed some of the analogies and some very powerful analogies too. I'm trying to think of my favourite. Yeah, much appreciated. I think would like to maybe do the wrap next time, if that's okay. So yeah, please do ask any questions. The last speaker is actually a video, so it might be useful to do questions now with the speakers if people would like to unmute perhaps and raise your hand. If not, I can read them out. Yeah, Andy, please.
Yeah, thank you. A question on prostate cancer. I'm a GP. Asked the other day by a man who had what I think has been labelled as a non-secretory prostate cancer. So he was diagnosed under the age of 50. His PSA was 1.5 at diagnosis. He's got metastatic disease, albeit subtly on bone scan and CT PET. And he asked the question, what should his son do? Does having a non-secretory prostate cancer have any? Obviously, the son will be at higher risk because this chap was diagnosed under the age of 50. Is PSA still a reasonable screening test in offspring, or is it more likely the offspring could have a non-secretory cancer as well, and therefore PSA may not be so useful? I think the man was diagnosed randomly at a health screening on prostate exam. Thank you.
Yeah, we don't see very many of these. We'd certainly recommend panel testing for him, which would include the BRCA genes and would include also the Lynch syndrome genes. We know that MSH2 is turning into a really quite aggressive prostate cancer gene as well. We take a family history as well, just to make sure there's not a neuroendocrine wider family history. PSA is not a perfect test. Classically, if we were to find a gene, then screening would start from 40. If we didn't find a gene, then probably it would start a little bit later, maybe at 45. But the advantage of finding a gene, of course, is his son may not have it. We do think in the longer run, PSA alone is not going to be the way forward. So his son will be a lot younger, but he needs to watch his space, because I would think that if there's a gene, you'd want to really include MRI here, particularly given that story with the low PSA. And the Royal Marsden is doing research for gene carriers, including MRI. And of course, the prostate cancer detection rates, because of that rule of 15 and the rule of 85, is remarkably high. But it is difficult to make sure you get it right with for instance, thyroid cancer. Over 90% is overdiagnosed. So you do need to be careful about diving in. So my advice would be, he's under 50 with metastatic disease, get to genetics, look for a neuroendocrine link, offer him a wider gene panel. Could consider tumor testing as well, because only half the gene changes are inherited, and see if we can find a target for therapy as well. But if there's no wider family history, there's a good chance you won't find a gene there.
Thank you.
Wait, any other questions? Stuart, are you still on the line, and would you like to ask your question? I can speak on Stuart. So Stuart was asking about in andrology, it feels like they do a lot of tests due to economic reasons, cost versus treatment. Let me it's looking at, I guess, how this might change in the future, and if it's and that's Joyce, can you see that question in the question and answers section?
No.
It's about basically he was thinking, like, how is it going to be possible to mobilize change when we have, I guess the IVFs and so on, which are quite economical these days.
Yeah. Well, it's interesting. At my, my inaugural lecture last Wednesday, I was asked, what would be the best genomic test? And I talked about, how genomics testing changed. But one of the things that has changed in genomic testing is actually the cost, right? So a genome now costs a few £100, right? So I can tell you one thing: a genome is not the problem. Of course, it's associated interpretation and all these kind of things. But I don't think that anything we do is very much to do with cost here. It's much more how you implement it in the system. So I think we have to somehow prove the value of this, and this is something that again, I mean, I'm a scientist. I've been very much involved in the Netherlands in the implementation of genomics for rare disease and cancer, and we've shown that it is extremely important in patients with rare disease, intellectual disability, and of course it's been taken up for so many diseases in the NHS now, and I think there's not a very difficult case to make to do this also for patients with severe forms of male infertility.
Do you think it's possibly more social changes that are required at this stage?
I mean, I think it is true that most andrologists don't seem to care, right? So I think the problem is that they just think IVF ICSI, and I think what they should do is do a quick genetic test. And if we can, it's not only about cost; it's also about quick turnaround. But if we can quickly turn around and we can tell them, well, actually, I'm sorry, but this patient has a mutation in the M1AP gene, ICSI will not work, right? I mean, that is already in itself, giving both a lot of benefits to the couple as well as to the cost saving. But I think this is something. I mean, I'm looking for help, of course, to implement this and get this into the NHS, and we'll see what we can do in the coming years. But anybody who wants to help is very welcome.
Great, thank you. Has anybody else got any more questions?
I can see you've got some Jedis from the Royal Marsden and the Institute of Cancer Research on the call, and Joe. Laura, I don't know if Joe wanted to say anything further just to finish off from our United Against Prostate Cancer project.
Just from me, if anybody wanted to reach out, we're happy to share resources of the work that we've done. I know that other areas are considering similar sort of projects going forward, and yeah, just sort of acknowledging the work that Julian has already mentioned, but the power of sort of the patient voice and going out and doing that outreach is paramount. So that co-creation piece of making sure that they are involved in sort of those initiatives and journeys is really quite powerful.
Thank you. Unless there's any other questions, I'm going to share the last presentation. I'm also going to put the link into the chat in case that's easier for people. So, sorry about this. Oh no, the computers are against me. Right, I have to do it the old-fashioned way, I think. So the next person who's going to speak is called Carl Lander. He's a registered nurse, and he also was himself diagnosed with a rare disease. And he does a lot. He's very passionate about patient advocacy and the role of the patient in obviously mobilizing the change. So I'm just going to scroll to his video now. And so sorry that this is taking so much longer, and I will of course put this in the chat as well if anybody would like to look at it in their own time. So in the meantime, right, here we go. So Carl Lander, as I said, registered nurse, patient advocacy leader, and he's going to tell you about his sort of experience here as a patient in this video.